The Science Basement!
Hello and welcome to the Science Basement podcast, a podcast for people who love all things science. I'm your host Rhiannon.
And I'm your co-host Oka.
Our topic today is something nearly everyone can relate to, the onset of puberty. Cast your mind back to your teenage years, to that uncomfortable time when the onset of puberty began and your body started to change. However, how does this process actually begin? And what happens if something goes wrong within this process?
To talk us through this episode is Nidhi Madhusudan, a PhD researcher in Raivio lab at the University of Helsinki. Welcome to the podcast, Nidhi. And how are you today?
I'm doing very well. Thank you, Uka, and thank you, Rhiannon, both for this opportunity today.
You're welcome. We're very excited to have you here. So let's start from the start, really. Most people will know of puberty and have experienced it, but what, in your words, is puberty?
Well, with puberty, it's, I think, both emotional and not something that most of us or any of us would want to relive. But physically speaking, for boys, it could be when their voice changes and hair starts to grow from their face, from their armpits, and when the testes drop. And for girls, it's when breast development starts and the onset of their period, which I think many women have either very fond stories of how it started or very uncomfortable stories of how this went about.
Yeah. So according to Inside Out, it's when anxiety, embarrassment, and envy suddenly comes into your life. Hello.
Perfect, accurate description of how a person feels during this phase.
Yeah. There's a lot going on at once. So in terms of the scientific side, how does this onset of puberty begin?
So a large part of my research is focused on the brain. And I think our audience would be fascinated to understand that this process actually starts in the brain. Mm-hmm. So we look at a very small population of neurons located in a part of the brain called the hypothalamus. And in the hypothalamus, these neurons are called gonadotropin releasing hormonal neurons, or GnRH for short. Mm-hmm. And GnRH neurons are the ones that start puberty or are the gatekeepers of puberty.
Okay, so these GnRH neurons are the neurons who are located in the hypothalamus that are really triggering the puberty to start.
Yes.
Okay, and how does this trigger happen? Or
that's the million-dollar question. And I think that scientists have spent years trying to understand. We still don't have the answer, is the short answer. What exactly triggers it? But we are getting closer into understanding how they even form or how they're not supposed to form. Mm-hmm. And I think the most fascinating thing about these neurons is that they're actually not born in the brain during fetal development. They're born in the nose.
Oh, wow. Okay. Mm-hmm. So the neurons themselves, when the fetus is developing, they start in the nose and then move to the brain because, yeah.
Yes. Okay. So basically during development, so when the fetus is developing, mm-hmm. The head will form with the brain and then you will have the nose forming. Mm-hmm. And the nose is one of the few structures in the brain that has a direct connection to the brain. It doesn't have like a complicated network of neurons, which are branching out. And so once these connections are made from the nose to the brain, the GnRH neurons have a road. They now have a track and they're like, okay, we see a direct connection into the brain. We're going to find our way there with this existing road.
Mm-hmm. Okay. So most neurons will start in the brain and then form links to the...
So when the fetus is in early development, it's not even really a brain. It's just several structures coming together, forming what will eventually be the brain.
Okay.
And you can also tell from the head and the spinal cord, or what used to be our tailbone.
Yeah.
You can see if you've ever seen any videos of the fetus developing. But each individual organ will also develop as a sum of its parts, basically.
Yeah. Okay, so the GnRH neurons are unique in the way that they first start in the nose and then
Yes.
move back to the brain, whilst most nerves or neurons go from the brain to their final destination. Okay.
So the neurons transmit to the brain when we are still a fetus or when we grow up?
This process happens actually very early in development, and this process happens simultaneously with the rest of development.
Mm-hmm.
It's interesting that it's a very
Early
early process, considering puberty isn't,
Yes.
Well, it's early in your life, but not 11 weeks.
Yes, not 11 weeks, no. It's quite the whole process is quite dormant until we actually reach adolescence.
Mm-hmm, mm-hmm. Yeah. So going back to the actual process in itself, so we don't know what is causing the trigger of the puberty, but what is the role of these GnRH neurons in puberty?
Okay. I'd like to introduce concept
Keyword.
called the HPG axis.
Okay.
So this is the hypothalamic pituitary gonadal axis. And a nice way to sort of understand this would be to think about the CEO of a company.
Mm-hmm.
And here the CEO would be the hypothalamus. And as I had said before, the hypothalamus is where GnRH neurons are located.
Mm-hmm.
So if you would think about the CEO sending out a memo
Mm-hmm.
to the company,
Mm-hmm.
and you think about the floor manager or factory manager of the company as the pituitary gland.
Okay.
He takes this memo and then gives it to his factory workers. And in this case, this would be the gonads. And for men, those are the testes. And for women, that's the ovaries. So if we look at this axis in its entirety, the hypothalamus will send out signals via the GnRH neurons, which then talk to the pituitary, which releases luteinizing hormone, follicle-stimulating hormone, and then this goes to the gonads, so the factory workers, which release testosterone and estrogen in our body. And I thought what was interesting is if you have an overproduction of these, so the factory workers are extremely efficient, the CEO will definitely hear back, and this is not real-world accurate, but he would normally, in the biological sense, your body would tell you to slow down the production of estrogen or testosterone, or say the workers are not working hard enough, then they would need to release more of this hormone.
Okay, so the hormone is kind of, in your analogy, these messengers.
Yes.
Yeah, that's causing moving the message from the brain to the gonads to cause the puberty, basically.
Yes.
Yeah. Okay, yeah. This HPG axis, that's the main
sort of system through which
yeah.
we look at these neurons.
Yeah. Okay.
I just wonder how puberty blockers affect this process.
Question.
Okay, so are we talking here then about trans people that want to sort of change or alter their gender?
Change their gender or just block the whole puberty process?
Okay, so many of them would, of course, then block these chemical messages that are coming, and these chemical messages are very sex specific, so what we call biologically male, biologically female. So if you were to block these messages, and then you would have to try to override the biological basis to reverse the process.
Mm-hmm. And then is it that you then can artificially supplement or take the hormone to initiate the puberty development of the alternative sex?
And I think this process is in itself, I think, both physically and mentally quite exhausting on the people.
Yeah.
Okay, so we've covered now the different kind of ways in puberty onset happens with the HPG axis and the hormones and different scenarios where this can take place. But what happens if someone's infertile? How do these GnRH neurons link to infertility?
Okay, so I'm looking at actually very specific kinds of infertility. Okay. And as I'd mentioned, the GnRH neurons are located in the hypothalamus, which is in the middle of the brain. So if you sort of find what is the midpoint of the brain, it's a very, very small area, and it's located very central. And I also mentioned hormones. So I'm looking very specifically at endocrine-related infertility.
Okay.
So when these GnRH neurons don't form correctly in the nose, or they don't find their way into the brain, they don't secrete the hormone that they're supposed to, even if they have found their way into the brain. There are many different things that can go wrong in this process, and I'm looking at these processes, and then if these things do go wrong, a patient is usually infertile.
Okay.
So this is basically a hormonal infertility.
Okay, so this is, does this mean that they actually don't even go through puberty in the first place? Or?
Exactly,
Okay.
exactly right.
So this is different to, like, you know, if you're infertile in a way that you might not know until
Exactly.
you try and conceive, for example, and then you
Exactly.
go through some tests.
And if we come back to the analogy of then looking at the factory workers, so maybe something is wrong with a person's ovaries, or something is wrong with sperm production and the testes. So this is much more downstream in the process.
Yeah, because
what-
the puberty's already happened.
Exactly.
It's just, yeah.
And this is sort of what my research is quite away from.
Yeah.
I'm looking at much earlier, and also looking at congenital effects, which are basically genetic mutations that affect the GnRH pathway and hypothalamus functioning.
Okay.
And for the mutation part, I just want to wonder, what if a child born without a nose?
It's a very good question, and a very interesting, and something that I would have never thought of. But fascinatingly enough, of course, if you're born without a nose and without any internal or external olfactory structures, your biggest problem will first be breathing. And you tend to have other comorbidities that may or may not even allow you to live. But those patients that are born without a nose, we find that, at least there was a study done, that most of the male patients were actually infertile, but the female patients weren't.
Oh.
Oh.
Interesting.
Which leads us to believe that females have potentially a lot of compensatory mechanisms to keep reproduction alive.
Yeah.
Like to keep the reproductive
Wow.
axis
What
stable.
can I say?
Yes.
It really is a woman's
Aw.
world.
Completely agree.
Wow, that's super interesting. So, yeah, I think these kind of models, or, well, scenarios where there's something missing from the regular system, you can learn a lot from.
Yeah.
How the in this case, how the neurons migrate from the nose to the brain. But if you don't have the nose developing, how does this happen? Then you know.
Especially in such a harsh context where a person is born without a nose.
Yeah.
So
Yeah. Okay, so you mentioned, like, your research, going back to before the nose, your research and the infertility. So what a problem in your specific research is it aiming to overcome?
all right. So as I'd mentioned again, this whole process of how these neurons go from the nose to the brain, there's many steps here that could go wrong. And we kind of know the path that these neurons take. We kind of know some genes that are involved in helping these neurons find their way. But we still don't know exactly how this process happens. So my research is focusing on the genetic environment and the molecular environment. So what does it really need for this process to happen?
Okay, and to do this in the lab, how does this work? Like, what techniques or what models are you using? Because, as you mentioned, this happens basically, you know, like in the fetus. So what kind of things are you using to study this?
Okay, in our lab, we actually have a stem cell model. And a stem cell here is basically a cell that contains the information to become any cell in the body. So if we look at it like a recipe book, it has the code for a recipe book. You're not ever making every single recipe all at once. So you will be making a specific meal, and the stem cell is like the recipe book for the whole body. And we have a model in the lab where we're actually the first lab in the world to do this.
Wow.
To make these GnRH neurons in a dish artificially. So we have the recipe to make the GnRH neurons.
Ooh. Good luck for your initiation.
Thanks. I was lucky enough to be here when this was already done. So I want to thank my
congratulations.
predecessors.
Ooh.
Yeah, that's very cool though, to be kind of leading the field in the
It's a very small field.
Yeah. Yeah, they don't need to know that. Okay, so you make the GnRH neurons from these stem cells.
Yes.
Correct. Okay, and why can't you use the GnRH neurons themselves? Why do you have to, in a sense, use the recipe to make the GnRH neurons?
It's a very fair question. And since we're kind of looking at how these neurons are born, if we take existing neurons that are already in their final stage, we kind of want to understand how they even develop. And we try to manipulate different processes during their development. So we need to kind of go back to what they were before they were the neuron, which is why we use stem cells as a model. A lot of other researchers have used mouse GnRH neurons because, of course, no one is going to take them from a human.
Yeah, exactly.
So
quite a lot of ethics around this, yeah.
Please cut my brain open.
Yeah.
Take what you would like.
Yeah, exactly. It's not going to go well. But yeah, okay. So you have to make the GnRH neurons from the stem cells to understand the process. Kind of remake this process in the lab. Yeah.
I just wonder, can't we take the new GnRH from a dead brain?
But that's exactly why the brain is already dead there. So we couldn't really assess.
Okay.
And also this, again, comes back, it's already in its final stage. We don't really, we want to know actually how it even gets to the final stage. So I think...
Yeah, so you have more control if you're making them, because then you can look at each stage. The steps of the stage.
So the point is to how it starts, not like how it ends. It's processing or how it ends.
Exactly, yeah.
Okay, so you use these stem cells to make the GnRH neurons. And what process are you using along this timeline to actually study the effects of the GnRH neurons, or what's happening along the way?
So we use a technique called CRISPR.
Oh, nice.
CRISPR.
But just put simply, it's a way to manipulate genes. So we published a paper in 2022 which sort of looks at which recipes from the cookbook are being used to make these GnRH neurons. So we have a list of genes that are sort of very important for these neurons, and we basically manipulate them. So we either remove them or we make more of one gene to see if we can either make more GnRH neurons or fewer GnRH neurons.
Yeah, so you have a list of genes that you know are somehow involved, and then by either removing them or adding more of them or increasing the expression of the gene, seeing how this affects the GnRH neurons to see how essential, I guess, it is in the process. Yeah. Okay, yeah, this makes sense. In biology, it's a very now common technique to use. It was, for those who don't know, it's like kind of, in the last 10 years, 5 years. Recently there was a Nobel Prize, but it's kind of revolutionized the field because it made it a lot easier to study how the genes basically affect the processes. Yeah, because in many ways, like in all sorts of biology, the genes are affecting the different processes of the body. So if you remove it, then what happens? How is it? Is it still happening normally, or is now something else happening? And
if you ask a geneticist, they will tell you that everything is genetic.
Yeah. Everything's related to the genes. They're the blueprint. Okay, yeah, and you mentioned this paper that your lab has published. What exactly did it find? Did you find in this paper?
Yes. So as I also said, that we have this protocol or this recipe that we use to make GnRH neurons. right now, in the beginning, our efficiency was quite variable. So in the brain as well, there isn't just one kind of neuron. There are several different kinds of neurons. But broadly speaking, we can categorize them into two kinds of neurons. So one are excitatory, so neurons that will make you do something, and the other kinds are inhibitory, so neurons that will make you stop an action. And in this paper, we found that GnRH neurons are the stop kind of neurons. They're more inhibitory. And we're also making several excitatory neurons or glutamatergic neurons, which was quite fascinating for us to see that our population of neurons is quite heterogeneous or quite mixed.
Okay, so even though you're trying to make just these GnRH neurons, you're actually making many different
kinds of neurons.
Okay, so it's not like 100%.
No, exactly.
Pure
or homogeneous culture is what we like to say. And an interesting thing there was, so as I said, our variation for this is quite high, which means if we have a good experiment, we can get maybe 10, 12, 15% of GnRH neurons. A bad experiment, maybe only 2%. So only
still
2
quite
cells.
low though, 10%.
Yeah. But we did manage to find with this paper a chemical that increases this efficiency almost 3 times.
Oh wow, okay. Yeah, because I guess in this process you're like adding the chemicals I think in a cell it's always hard because you don't have the whole body, whilst in the you have the whole organism that's normally happening, so you're kind of trying to recapitulate this but in a dish, so you're like educatively guessing what chemicals to add.
Exactly.
So, yeah.
But I think the other interesting thing to note is in the actual human brain or in the whole body, there is a delicate balance of everything, and no matter what we do, so there's obviously a reason that we're only able to make 10%, 12% of neurons. There's going to be a threshold beyond which we're not going to be able to make any more because they're a very small population compared to the billions of neurons. So, of course, even if we're trying to do this artificially, in some sense, it's quite validating for us to know that even when we do this artificially, we're still only able to sort of recapitulate what the body or what the brain
because 10% sounds like a little, like a small amount, but when you say it like that way, then because I guess from the stem cells, really, even though you're adding all these chemicals, you can't specifically make only the GnRH neurons. So in this case, these chemicals are still making many types of neurons as it would happen normally.
Exactly.
In the fetus when it's developing.
Exactly.
Okay. Yeah, that's very interesting.
Actually, I might go a bit off topic now, but I'm still wondering, how did you identify that GnRH hormones are inhibitory?
GnRH neurons are inhibitory because they express the genes that are inhibitory. So if you have to describe how this neuron is, we have certain genes that we know that all inhibitory neurons will share, whether or not they're GnRH or not.
So like a signature.
Basically.
Kind of,
Basically.
So these habits are such as...
Okay. Some genes are GAD1 and GAD2 are two very common inhibitory neuron marker genes. And similarly for excitatory neurons or glutamatergic neurons, it's quite easy. They're just called GLUT markers.
Yeah, that's easy. Sometimes genes are very logically named, and sometimes it's completely random, and then you're like, where is this?
GnRH neurons are also called GABAergic, so they're also called GABA markers.
Okay, yeah. Nice, yeah. That makes sense. Then it's, because I guess like even in my field in cancer research, we have certain markers that marks, well, not a cancer cell actually, because that would be too easy. Then we could easily make treatments because it would target just those cells. But for example, certain genes that are linked to the cancer specifically. So normally in these sense, like all cells will have at least
identity.
One gene, an identity that makes that cell, and not like a liver cell or a stomach cell, so
or a heart cell, like
one
of the main things to identify heart cell is if it pumps.
Yeah.
One of its main functions is to pump.
Mhm.
And if the cell is pumping, it's most likely a heart cell.
Yeah.
So-
Yeah, exactly.
Huh.
Yeah.
Interesting.
Yeah. But yeah, so that's cool. So you found... So did you then find some genes that were linked to the inhibitory...
Trajectory.
Yeah.
Yes.
Okay.
Yes, and that's basically what we will be doing in our future studies.
Okay.
We found a very nice list of genes that we could look at
Mhm.
and varying aspects of these genes and how they then influence how these GnRH neurons are formed. Do they even influence how the GnRH are formed? Maybe it's just a coincidence.
Yeah. Yeah.
With biology, nothing is ever
Simple.
linear. I just want to say
Easy, linear, or simple.
Yeah.
But that's what makes it very interesting, actually, because you think, oh, okay, I have this hypothesis, this is how it probably should work. And then often it's, I mean, the human body has a very...
It's very beautiful.
It's very, it's amazing in the way that it can adapt and, you can remove something and then it still can function, and then it's very interesting because you think, oh, this is essential, and no, it's not. Or, yeah, so okay, that's very interesting. So now you're looking into further how these inhibitory genes signature causes the development of the GnRH neurons. Okay. And in this process of going back to the technique of from a stem cell to GnRH neurons, how long does this take on average? How does it look in the lab, kind of day to day?
So for our protocol, from day 0 stem cells, with the cookbook, to GnRH neurons, it's 25 days. So it's almost a month.
25 days.
Yeah. But think about it, the fetus is developing 9 months. So for us to get these
That's
functional
true.
neurons within a month, it's already, it's faster compared to several other neuron protocols, but it's still longer than most other protocols.
Yeah. And this is quite a intensive, like labor-intensive process. Right?
Yes. The one thing that does help us is that the default sort of cell that a stem cell would want to become is the neuron.
Okay. Oh, interesting. So it can become any cell, but it prefers to
become the neuron. I think the brain is sort of... You kind of need the brain to start and initiate all the other processes anyway in the body. So it makes sense that the default would be the neuron.
Yeah. Yeah.
You need the control center.
Yeah. Especially when you're developing. I mean, if you can't form the brain, then...
Exactly.
Yeah. Oh, interesting. Makes a lot of sense, actually. Yeah, so then in this 25 days, you're always having to look after the stem cells and...
They actually need to be fed every day. My mom
Fed
keeps
every
asking me
day?
how the grandkids are.
Oh my god, how do they feed?
So since we work in vitro, so basically it's an artificial system, we have to feed these stem cells media, or basically it's a liquid that we give them with all the nutrients they need to grow.
Yeah, it's like the meal of the day.
Yeah. And they need to be fed on a 24-hour cycle. So for 25 days, we need to go in every single day to feed them, keep them alive.
That's why you're calling them babies.
Yes.
Because they need a lot of attention.
Yes. A lot of very intense attention.
Yeah, so you need to be very careful under a high control responsibility to do those lab works and like take care of the stems. That's
Yes. They need to be under very sterile conditions, because we need to remember this is outside the body. So inside our body, everything is for the most part sealed, whereas we're doing this outside, externally, on plastic dishes, sometimes glass dishes. So we need to sort of replicate the conditions as similar to the human body as we can.
Yeah.
Okay, so then how do you get those stem cells?
Basically, they're commercially available from a company. You can actually anyone can just order them to the lab. Of course, you need the correct licenses and the permissions to be able to use them. But you can basically just order them off a company, and someone donated their stem cells, I think in the early 1980s or 1990s, and this is something that labs all over the world are using for their experiments.
And is it the expenses of the reagents you use, is it kind of more expensive than other labs?
We have been lucky as PhD students in the lab to be kept in the dark
Yeah.
about a lot of the finances. But also, to be honest, one of the chemicals that we use costs €1,000 for 1 ml.
Oh my god, yeah.
Of the chemical, and 1 ml would maybe last me alone 3 months.
Okay. So it does last a while, but still, yeah.
But we're normally like 5 people in the lab, so then we say it's maybe a month.
Yeah, yeah.
So
Yeah, that's expensive.
Yeah, that's expensive. But from which part of the body do you get the stem cells? Like, it can be from any part of the body, or
so this is a very good question. There was a Nobel Prize in 2006 by Yamanaka and Takahashi, who managed to make any cell in the body back into a stem cell because they knew the recipe for what a stem cell is. So originally they could be taken even from the blood, which is easily accessible. Before this, a lot of the studies were done in cord blood, so umbilical cord blood stem cells. And this was, of course, as soon as the baby is born, they would take the cord blood cells, and then we have the stem cell banks where you keep the baby's cord blood. And this is still something that is done now. If people want to keep their child's stem cells, it's not so much needed because we know how to make the stem cells. So you could take anyone's blood, an adult even, and reverse it back into a stem cell.
Yeah, it's pretty cool. Going back to your research itself, what's the next steps you're looking to do, or what are you working on at the moment?
We're actually fascinated by something that happens around the GnRH neurons. So further deeper into the brain, something that actually unlocks the GnRH neurons themselves.
Okay.
So
What do you mean by unlock?
So sort of triggers their response. There's actually someone even higher up than the CEO.
There always is.
And they have a very fascinating name. They're actually called KISS neurons.
Okay.
KISS neurons?
Yes, and they're named after Hershey's Kisses.
Oh, really?
Yeah.
Oh, this is a classic biology thing.
Of course, because the scientists that discovered the receptors for these are from Hershey, Pennsylvania.
Okay.
And they named the neurons KISS neurons after the
funny.
chocolate.
And it's ironic that they're also involved
In puberty.
in puberty.
Exactly.
Which is
come
often
full
linked to
circle.
kissing.
It's come full circle.
Oh, that's funny. Okay, so what do these KISS neurons, or cells, neurons, what do they do, or what have you been looking into regarding them?
So what do they do is basically they have the key to unlock GnRH neurons. And our lab is just, again, studying.
Okay, so you're trying to, it's basically like you're doing the same thing, but a bit earlier in the process to work out what is actually causing the GnRH neurons to start the puberty.
Yeah.
Okay.
Exactly.
I guess we can look out for some research that might be coming out soon to find out how you look into these, into the KISS neurons and things around this.
Yes, stay tuned.
Stay tuned to find out. Okay, very cool. I think we've covered very interesting, really the molecular biology side of puberty, which I, didn't really know. As we mentioned at the beginning, everyone knows what puberty is and everything, but it's not really very common knowledge. Maybe the hormone side of things is more known, especially everyone. Maybe more for women than men, but most people know the hormones affected in puberty and in kind of reproduction, but not exactly how it starts. So
I think we all also know about testosterone supplements.
Yeah,
this kind of
true,
thing in the
yeah.
sports field.
Yeah.
So
yeah, true. There's very overlapping in this side, but I think this kind of earlier stages is very, at least in relation to infertility itself. You don't often think about people who don't go through puberty. Maybe just, as we mentioned, people who happen to find out they're infertile later on in life. So yeah, thank you for this. Before we end the episode, do you have some take-home message that you would like to give to the listeners?
Yes, actually. I think we didn't really talk about the real-world applications of this research, and I tend to think sometimes scientists are in such a deep rabbit hole that they don't really realize what the larger implications are. And I think especially in research that's so developmental and early stage, I think it's sometimes hard to miss what is the point of studying all of this. And it's actually, I think, a valid question because we're not yet in a phase where it's like The Handmaid's Tale. You know, infertility is not as scary as cancer or Alzheimer's. It's not fatal either. So it's not really, in quotes, a real world issue. But I think what my research is really helping is to give people a choice on whether they want to have children or not. Because most of us, I think, take this aspect for granted. And we don't know anything about patients suffering from infertility, and especially this specific kind of infertility. And I think my research would one day give people the choice back.
Okay, so I think that's all for today, this episode. Thank you very much to Nidhi for taking the time to join us at the Science Basement podcast today. It's been super interesting to learn, as I mentioned, all about your research and the GnRH neurons and into how they link to the onset of puberty. And I hope you enjoyed the discussion today.
Thank you very much for having me. And I learned quite a bit as well.
It always helps to communicate your science and then you realise,
Nice to hear that.
That's a good question. And as a reminder, keep looking out for our new episodes on whichever the podcasting platform of your choice. I've been Rhiannon.
And I've been Oka.
And this was the Science Basement podcast. Thanks you all for listening and see you in the next episode. Goodbye.
Bye.
Bye.
The Science Basement.